Today, we're going to talk about one of the most ambitious scientific quests, maybe ever, cracking the actual code of life itself, the Human Genome Project.
So just think about this for a second. What if we had the complete blueprint for a human being? I mean, the full instruction manual for everything, from your eye color to your risk for certain diseases. What could we learn? What could we fix?
For the longest time, that was pure science fiction, right? But then, in 2003, it actually happened. Science delivered. And this is the incredible story of how they pulled it off. So, where did this all start? Well, it began as this just unbelievably audacious quest. A project so big, so ambitious, that a lot of people were pretty sure it was impossible. And when I say ambitious, the scale of this thing is just, it's hard to wrap your head around. The goal? Map the entire human genome. We're talking about 3.1 billion of these tiny chemical units called base pairs. Think of them as the letters in our genetic alphabet. To put that into perspective, if you printed that whole sequence out, you'd have a stack of books 200 feet high. Just incredible. Okay, so what was the Human Genome Project officially? Well, at its core, it was this huge international research project. Scientists from all over the world were working together with two main goals. First, figure out the exact order of all those billions of letters. And second, find all the actual genes, the instructions hidden inside that code. So, things officially kicked off in 1990. It was led by the U.S. Department of Energy and the National Institutes of Health, and they gave themselves 15 years to do it. A really big moment came in 93 when Francis Collins came on board to lead the whole thing. And here's the kicker. They actually finished in April 2003, a full 2 years ahead of schedule, which, for a government project of this size is pretty much unheard of. So you're probably wondering, how in the world did they even start a task that big? Well, the public project had this really smart methodical plan. They called it the hierarchical approach. Think of it like this. Instead of trying to read a giant 3 billion letter book all at once, first they broke it down into large chapters. Then they figured out which chapter belonged where, mapping them to our chromosomes. Then they read each chapter individually. And finally, they put it all together into the full map. It was slow and steady, but it was designed to be super, super accurate. But just when things were moving along, the whole story got this massive jolt of adrenaline. What started out as a friendly global collaboration suddenly turned into a crazy, high-stakes race. And this race had two very different teams. In one corner, you had the public project, this big international group with a nearly $3 billion budget. Their whole philosophy was science for everyone. They released their data for free every single day. And in the other corner, a brand new private company, Celera Genomics. They were led by this brilliant, kind of controversial scientist named Craig Venter. Their budget was way smaller, but their goal was all about speed and, profit. They were using a totally new method and planning to patent the genes they found. Yeah, Celera's strategy was a complete 180 from the public plan. Instead of that careful chapter-by-chapter approach, they used something called whole genome shotgun sequencing. And it's pretty much what it sounds like. They basically blew the entire genome into millions of tiny pieces, sequenced all those little bits at the same time, and then used these incredibly powerful supercomputers to try and stitch the whole puzzle back together. It was a massive gamble, a total high-risk, high-reward shortcut. You see, this wasn't just about who could sequence faster. It was a battle of ideas, a really fundamental question. Should the blueprint for a human being be owned by a company, or does it belong to all of us? The race got so heated, it went all the way to the top. I mean, you had the US president and the UK prime minister issuing a joint statement, basically saying this information has to be freely available to everyone. That sent absolute shockwaves to the biotech world. So, who won the race? Well, it's actually not that simple. As it turns out, there wasn't just one single finish line. Yeah, in science, the word complete can be a little tricky. Here's what happened. In June of 2000, with tons of pressure on both sides, the two teams basically called a truce and announced a working draft together. Then, in 2003, the public project announced their version was complete, but believe it or not, the story wasn't over. For years, as the tech got better, scientists kept chipping away, filling in the missing pieces. And it wasn't until, get this, 2022, that a different group of scientists finally published the first truly 100% complete, gap-free sequence of our chromosomes. So what did complete actually mean back in 2003? Well, it meant they had successfully mapped about 92% of the genome. And that was the most important stuff, the parts with most of the genes. But that still left about 8%, these really tricky, repetitive, super hard to read sections that were still just gaps in the map. So it was an absolutely world-changing achievement, for sure. But it wasn't quite the final word.
Okay, so with all that history, let's get to the big question. Why does any of this matter? What's the real legacy of this incredible project?
I mean, having this map has completely changed the game for medicine and biology. It's let us finally understand the genetic roots of diseases like cancer. We can design brand new medications based on someone's specific DNA. And it's not just medicine. We're talking huge leaps in forensic science, new insights into human evolution, even better ways to grow our food. It literally changed the way we study life itself.
And you know what's really remarkable about this project? The foresight they had. Right from the very beginning, they knew that this powerful new knowledge would open up a whole can of worms, all these tough ethical questions. So they actually set aside a big chunk of their budget, 5%, just to study the ethical, legal, and social implications, or ELSI. So while they were cracking the code, they were also asking, okay, how do we handle this responsibly? What about privacy? What about discrimination? That was just brilliant.
And that really brings us to the final thought here. The Human Genome Project didn't just give us a bunch of answers. It gave us a new language. It turned our own genetic code into an open book that we can now read. So the real question, the one for all of us now, is what are we going to write in the next chapter?